Okay, good morning, everyone. My name is Paula Clemens. I'm a professor of neurology at the University of Pittsburgh. Have devoted my entire career really to studying Duchenne muscular dystrophy and working toward treatments. And today I'm speaking on behalf of a sponsor, NS Pharma, who has a drug product in a clinical trial, and I'm study chair for that multi-center study. So just a couple of disclosures and disclaimers, and just to indicate that I don't have any real or apparent conflicts of interest with this project. This is an overview of what I'm going to tell you over the next few minutes. First, I'm going to tell you a little bit about the sponsor and the partners that are involved in this clinical program. I'll tell you about the investigational drug, which is an Exon 53 skipping product. Tell you some about the clinical trials that have taken place with this and those that are ongoing. Give you some of the latest updates and the next steps. So first I want to introduce you to some of the partners, and that includes the sponsor, NS Pharma, a consulting company that's working with them, PharmaLex, and also the Synergy Network and its coordinating center, TRINS, and I'll introduce that in the next couple of slides. So the sponsor parent company is Nippon Shinyaku. It's a Japanese pharmaceutical company that's about 100 years old. It has a number of worldwide offices, including a subsidiary in New Jersey, which is called NS Pharma. So NS Pharma has teamed together with a consulting company, PharmaLex, to administer to sponsor their trial. And then they have also coordinated with the TRINS CRO, which is a muscular dystrophy specific CRO for doing clinical project management, and is also the coordinating center for the Synergy Clinical Study Network, which is a network of about 25 academic sites worldwide that collaborate together to do muscular dystrophy research. So NS Pharma has a program in Duchenne muscular dystrophy, which currently is focused on its most advanced studies, which are to study this Exon 53 skipping. I'm going to tell you primarily about the studies ongoing in the US, but also mention a parallel study going on in Japan. But in addition to that, they have preclinical and discovery programs in other exon skipping areas. So first about the investigational drug. It's an antisense oligonucleotide. You've heard about a number of these. This one is, like a couple of the others, is based on a morpholino backbone. This product is a little bit different and has been, the biochemistry of which has been developed in Japan. As I mentioned, it was in Nippon Shinyaku is the company, and they work together with a academic collaborator at the National Center of Neurology and Psychiatry to develop this investigational product. Its mechanism of action is to skip exon 53, and it's delivered as an IV infusion given once weekly. At this point in the conference, you know a lot about exon skipping, but the concept is, of course, basically to change the exon components to take an out-of-frame deletion into an in-frame deletion by skipping one extra exon at the transcript level. And that allows the body to produce a partially functional protein that retains both the amino and carboxy ends of the protein, but is missing a segment in the middle. So Shown here in yellow, you see the set of exons or the set of deletions that could be approached by exon 53. So in hearing about different drug development here over this weekend, you've heard about those that are mutation specific and those that are mutation non-specific. And this falls into the category of one that's mutation specific. So it's only applicable to patients with deletions that would be brought back into frame if you added an additional deletion of exon 53. And as you can see here, that by looking at databases that have been developed over many years, this could potentially approach 8% of the Duchenne population. So first I'm going to tell you about a phase 1 study that was completed. This was the initial safety study that was done at a single site in Japan with 10 boys with DMD. And it started at it's what we call a dose escalation study, starting at a very low dose and working up through 2 subsequent doses, each cohort of 3 to 4 patients being treated with 12 weeks of treatment. And this is an initial safety study that allows one to take a new drug product and look to make sure that at the lowest doses that it will be safe. Just for the sort of high level findings from this study, the first was probably the most important, which is that they demonstrated what is referred to as target engagement. So the whole idea of target engagement is that you introduce a compound that you expect to have a certain biochemical effect, and then you demonstrate that there's evidence for that biochemical effect. So they were able to show that by introducing this product that they could see evidence that exon 53 was skipped. At the very highest doses, they began to see evidence of some dystrophin protein expression, which is very encouraging. And very importantly, there were no serious adverse events during this study, nothing that caused any discontinuation of treatment. So now to the current study. So this is a study that's ongoing in the United States. It's a phase 2 dose-finding study, which continues the work that was begun in the phase 1 study. Again, its primary emphasis is to assess safety, tolerability, and pharmacokinetics, which is simply looking at the drug levels in the blood after you introduce a drug and how long it lasts there. It's also looking at pharmacodynamics, so that's the effect on other proteins in the blood when you introduce a drug. There are a number of sites across North America that are enrolling participants in this study. And now what I want to do is just walk you through the design of the study. This is also a dose escalation study with 2 dose levels. One dose level is 40 milligrams per kilogram. The second dose level is 80 milligrams per kilogram. So if you start over on the left side of your screen, participants enter into this study through a screening phase where it's determined whether they meet the inclusion and exclusion criteria. And once that happens, they're randomized into the study and have an initial pre-treatment muscle biopsy performed. And at that point, they are then randomized for the first 4-week period where they will either receive the investigational product for 4 doses or placebo. And the idea for this is to have a blinded, placebo-controlled initial safety phase for the first 4 weeks that allows a proper assessment of that acute safety from the investigational agent. At the end of the first 4 weeks, all participants, including those that were initially on placebo in the blinded phase, go on to open label drug for an additional 20 weeks. And then at the end of that first 6 months of the study, there is a second muscle biopsy to look for dystrophin production. And at that point, participants can then either exit the study or continue on into a long-term extension study. So the important point is that all participants in the study do receive drug, except during that first 4 weeks when a subset received placebo. There are 2 muscle biopsies as a part of this study. And the initial 40 milligram per kilogram dose is demonstrated to prove safe before moving to 80 milligrams. And if you remember back to that phase 1 study, that went up to a top dose of 20 milligrams per kilogram. So the concept here is to continuously increase it as it's shown safe. So in each of the 2 cohorts, there are 8 participants. They are between 4 and less than 10 years of age when they enter the study, and they need to have a mutation that would be predicted to be benefited by exon 53 skipping. So inclusion criteria, as you know, are the way that study designs create a sufficiently homogeneous population that the outcome measures will be meaningful. And similar to other approaches where the attempt is to restore dystrophin, we believe that it's plausible that it makes the most sense to initiate those kinds of therapies as early as possible. Hence, for this study, the inclusion age is 4 to less than 10. And patients need to be able to independently walk, and they need to be able to do the studies that are part of the outcome measures. There are also some key exclusion criteria, which are basically designed to make sure that it is safe for the participants in the study. The study endpoints, the primary ones are safety and tolerability. Safety is always the top concern in studying a new drug, but the efficacy, the primary efficacy outcome for this study is a biochemical surrogate outcome, which is that muscle dystrophin protein. And then because this is very early in drug development, there's interest in looking at the pharmacokinetics of the drug. There are many things that are being studied as well in secondary endpoints, including both the transcript and other measures of dystrophin protein, as well as all the strength and mobility and functional aspects of outcome. So the first cohort at the 40 milligrams per kilogram is fully enrolled, and those initial patients, those initial participants have completed, all of them have completed that first 4-week blinded interval. And so at that point, the study protocol had a DSMB review, which happened, so DSMB stands for Data Safety Monitoring Board, had their review a week ago to review that safety data from cohort 1 and to make the determination of going on to cohort 2. There were no serious adverse events. You can see kind of a listing of them to the right of the slide. Of all you see a listing of all of the adverse events. There were none that were serious, none that were drug-related. All were mild. None led to discontinuation of the drug, and the Data Safety Monitoring Board determined that it was recommended to go on to the second cohort of the 80 milligram per kilogram dose, which is just starting now this month. I just wanted to briefly mention that there is in parallel a unrelated study ongoing in Japan, which is of a similar design. There are some differences in terms of inclusion. The age of inclusion is wider in the Japanese study. I'm not personally involved in the Japanese study, but wanted to indicate that it is also ongoing, collecting data, and so far has also not had any concerning safety signals. So I just want to finish up with next steps for the U.S. study. As I mentioned, the study's been approved to go on into enrolling cohort 2, and that dosing will start this month. And a full complement of participants have been identified, so that will enroll quite quickly, and we will hope to have data from this study by 2018. There is an extension study, so some participants are enrolling into the extension study now and will be progressively as they finish the parent study. And we'll continue to collect safety endpoints as well as functional endpoints. And with that, I will complete and let the next person.